All right. Welcome to this lesson on stems. Throughout this video, we're going to be exploring some of the external anatomy of stems, some of the internal anatomy of stems, a little bit about stem physiology, and also some specialized modified stems that plants use to aid in their survival in different environments. Um, Regarding the internal anatomy of a stem, I'm probably not going to get really in depth in some of the nitty gritty, different types of cells and like different types of ground tissue. So if that's the kind of video you're looking for, this might not be the one. Um, we're going to try to keep the focus on kind of horticulturally significant um, stem anatomy. Um, yeah, so here we go. Okay, so what are stems to begin with? A stem or a shoot, those are one of our major plant organ systems, along with leaves and roots and flowers and fruits and cones and the like. Um, Stems typically have a couple primary functions. One is to transport materials between the roots and the leaves. If you can find leaves on a plant and you can find roots, that means there has to be a stem connecting the two. We never see leaves directly attached to roots, and we never see roots directly attached to leaves most of the time. I will, I guess, walk that back and say that sometimes things like tomatoes can actually develop roots out of the veins of their leaves if they're touching the ground, but that's not super common. Um, so they typically serve to transport materials between the roots and the leaves. They also serve to extend and direct plant growth. We often think of this as directing plant growth upward, but that is not the case for all stems. Many stems direct growth horizontally or even downward. And the stems also support the leaves. Stems can also be modified to have a huge range of specialized functions, which we will be discussing um, in the last third of this video. Okay. External stem anatomy. So we're going to be looking at the outer parts of a stem first, and then we're going to be looking at what's happening inside the stem in a minute. Okay. So stems are composed of several external parts: nodes, internodes, axillary buds, apical buds, and an epidermis. We'll discuss each one of these piece by piece. The first and possibly the most important part of a stem is a node. So a node is just a part of a stem where a leaf is attached and from which new structures such as buds, stems, leaves, flowers, or roots can grow. Nodes are usually pretty identifiable on a stem. They're a place on a stem where something is happening. And they're often marked by line, scar, swelling, the attachment of a leaf, the presence of a bud. If there's something happening on the stem at that spot, it's probably a node. Um, and nodes are significant because all new development occurs from a node. You can't grow new tissues from a place that isn't a node. The internodes are simply the spaces between the nodes, and unlike nodes, they are not very good at producing new structures like buds, stems, leaves, flowers, lateral shoots, etc. Um, so yeah, pretty simple. Internodes, just the spaces between the nodes, and they don't typically do very much. They do, though, have a function still. You can imagine that if all the nodes were packed together very, very tightly, there wouldn't be enough space between the leaves for them to really get enough light. The leaves would self-shade a lot, and a plant does not want to be self-shading. So part of the reason plants have elongated internodes is to space out their leaves so that the plant doesn't shade itself. That way, it's kind of getting optimal light exposure on each leaf. And immediately, nodes and internodes, or knowing about nodes and internodes, is super useful in horticulture for the preparation of stem cuttings. So, if you've never rooted a cutting, they're sections of stems that you cut from a plant, and they're used to vegetatively propagate or clone the plant. So you just cut off a piece of stem. You might expose it to some treatments like rooting hormone, or you might just stick it in a glass of water. You might stick it directly in potting mix, and it'll grow roots, and you'll have a new plant that's genetically identical to the plant from which you took the cutting. Because we can only get new growths of like buds and shoots, etc., from a node, it's absolutely essential that every cutting have at least one node. Without a node, we typically don't get good root formation because that occurs better from a node. We also typically don't get good shoot development of new shoots because once again, we have to have a bud at a node in order for that to occur. So you never want to take a cutting that is just composed of an internode. In fact, it's much better if every cutting both begins and ends with a node in most cases, because those internodes, after they're cut, they tend to like to kind of die back to the nearest node anyway. Um, Moving on, axillary buds. These are also called lateral buds. These are buds found at each node along the stem in the axil. And the axil isn't so much a part of a plant as it is kind of a region. It's that upper angle made between the leaf and the stem. So that comes from the Latin word axilla, which means armpit. And if you look at that upper angle between the stem and the kind of the top of the leaf, you can see that it is this acute angle, less than 90 degrees. It's kind of like an upside down armpit. And the axillary buds are tucked right into that armpit, as it were. Um, those axillary buds are going to then develop later into lateral shoots or side shoots, or sometimes you might call them lateral branches. You can kind of call them whatever you want as long as it's adequately descriptive. Lateral shoots, side shoots, lateral branches of a plant. Often the axillary buds may also develop into an inflorescence, which is something else to consider. It depends on the species. And then our last type of bud that we'll look at will be apical buds. These are also called terminal buds. And, a terminus is an ending of something. To terminate your employment would mean to end your employment. The terminator, he ends, I suppose, human lives. So terminal means the end, right? So the apical or terminal bud is the bud at the end of the stem. And the word apical comes from the same root word as we get apex, which means the kind of the tip or the top. So like an apex predator is at the very top of a food chain or the food pyramid. Um, so apical buds, terminal buds, those are synonyms. They're just the buds at the tips of the stem, and they extend the growth of the plant. You'll notice I don't say extend the growth of the plant upward because sometimes they're going to be extending the growth of the plant horizontally or even down. So it really depends on what type of stem is doing the growing. But apical buds do kind of lengthen the stem. When we remove an apical bud, which we do all the time in horticulture, we call this pinching, and it basically induces axillary bud growth. It kind of forces those axillary buds to develop and sprout and begin to form into lateral branches. Why? This is a really kind of important question in horticulture because pinching is something that occurs all the time in greenhouse production and nursery crop production. I think it's important to understand the hormonal physiology of the pinching and branching process. And to understand, we've got to understand auxins. Auxins are plant hormones which are produced by the apical bud. And I mean, you could spend an entire PhD studying auxins and still not know everything there is to know about auxins. Auxins are an extremely important plant hormone that does a big range of things. Excuse me. Um, to just look at a few of them, I've got a list here of things that auxins do, um, but I'm going to focus on just the highlighted ones. Some of the most kind of horticulturally relevant things that auxins do is they suppress the growth of lateral shoots or the growth of those axillary buds. So when you look at this Lebanon cedar tree on the slide, you can see that the central shoot of the tree, the trunk, that primary stem is much thicker and much better developed and has grown much longer than any of the secondary or lateral shoots. And that's because auxins produced at that apical bud influence and suppress the growth of those lateral shoots which is a process we call apical dominance. Apical dominance is basically the observation that the apical bud is in charge. It's the one that grows the most, and it suppresses the growth of the lateral shoots because it produces auxins. Um, Another thing that auxins do that's very important in horticulture is they regulate phototropism. Phototropism is a behavior of stems where they will grow toward a light source. Leaves will do this too, but in leaves, it tends to be that the leaf will tilt its lamina or its the leaf blade toward a light source. With stems, the tip of the stem, that apical bud, will actually grow toward the light, and that's basically because auxins are destroyed by light. So you can see in this diagram here that auxins are produced in the apical bud, and they move, they flow downward through the stem, and they are destroyed by light. And because auxins promote stem elongation, um, they kind of accumulate on the dark side of the stem where they're not being destroyed by light, and they actually cause the stem to elongate more on that side, which is very relevant. If you've ever left a plant in a windowsill for too long, you'll notice that it'll start to press itself up against the glass, and it'll lean entirely against the glass of the window because that's where the light is. The lights inside your house typically are still darker than any amount of sunlight that is available outside. So plants tend to want to grow towards the window. This is something that we need to be aware of in horticulture when we are spacing greenhouse crops or nursery crops. If they're packed too tightly, they will want to grow toward the light, and sometimes they will become kind of misshapen, or they'll lean one way if we have an uneven light exposure. So it's important to make sure that our light exposure is as even as possible all the way around the plant. Auxins do one more very, very important thing. Um, I've kind of briefly mentioned that the auxins are produced in the apical bud and that they flow down the stem very briefly. I mentioned that. Uh, they do go all the way down to the roots, and they actually promote root growth at the root tips. And we can take advantage of that function of auxins to root cuttings. Um, if you've ever used rooting hormones such as Hormodin, or another good brand is Dip and Grow. There are more. Clonex is another one. If I didn't say your favorite brand, it's not personal. I don't have any brand deals with any of these companies, um, but they all are based on auxin rooting hormones. And the most common rooting hormone that's used in the horticulture industry is indole 3 butyric acid, or IBA. So if you buy a container of any type of rooting hormone, be it a powder or a liquid, indole 3 butyric acid is almost always the main active ingredient. Plants more commonly produce indole 3 acetic acid, but indole 3 acetic acid is not particularly shelf stable. It tends to break down pretty quickly. Indole 3 butyric acid has basically the same effect, but it's shelf stable. And then another auxin that you see sometimes in these formulations is naphthalene acetic acid, and that one isn't, I would say, as commonly found in rooting hormones, but it certainly can be used in conjunction with indole 3 butyric acid in certain rooting hormones, especially for plants that maybe are a little bit more challenging to root. And like I said, auxins have a huge impact on apical dominance. So apical dominance is basically the suppression of axillary buds that is achieved by auxin production in the apical bud. So once you remove the apical bud, you stop or terminate the production of auxins, and that actually liberates all of the axillary buds to grow. So if we want to produce a bushier plant, it's very easy to just pinch off the apical bud, and then your plant branches out, and that can produce a much more attractive plant. In some cases, you wouldn't want to do that with conifers such as pines, because we actually want them to have a nice, strong dominant leader. Um, A lot of cut flower producers also do this because you get kind of a more well-branched plant with more weight near the bottom, so it's less likely to lodge or blow over. And you get a greater number of marketable stems throughout the growing season. As you can see on this zinnia, our flower grower pinches back the stem, the axillary buds begin to develop, and it produces a much larger, bushier plant. The epidermis is the skin of a stem, same as our epidermis. That's just the outer layer of skin. And that just protects the internal stem against damage, infection, and water loss. The same things that human skin accomplishes for us. One of the unique features of plant epidermises is they frequently have epidermal appendages. So these are little outgrowths of the skin of the plant that can be used for a variety of purposes, often defensive, but sometimes they're related to things like temperature regulation. So one of the first and kind of most familiar for all of you will be prickles. If you've ever been stuck by the prickles on a rose, you'll be very familiar with those. A lot of people would refer to them as thorns, but thorns are not the same as prickles. So botanically speaking, roses do not have thorns; they have prickles, and these are just thorn-like outgrowths of the epidermis. Um, True thorns are actually a modified lateral shoot, and the prickles are not actually a stem; they're just an outgrowth of the skin of the plant. And then trichomes, which are little glandular hairs. So they're not. I should clarify that they're not always glandular. Sometimes they are producing some kind of substance, and sometimes they're not. But they help protect the plant from a variety of different things. And on this slide, we can see some examples of prickles on this rose stem. We can see, and then two different types of trichomes. The one in the middle is a species of mint, and you can see that there's this dense down of trichomes. And I would assume that that probably has something to do with defense from insects. Some species actually have really, really, really dense layers of trichomes or little hairs. And sometimes that dense silvery white coating of hairs actually helps reflect light and heat and helps plants tolerate extreme heat stress in environments like deserts. And then on the far right-hand side of the slide, we can actually see the stem of a stinging nettle. And stinging nettles are really unique because they have hollow trichomes in which they produce venom. And you can imagine that if you're an insect, that's probably not going to deter you because you can just waltz right between these spines. But if you are an herbivore like a deer or a human being, those are going to be very painful. The trichomes of stinging nettle are reinforced with silica, which is silicon dioxide is a really common soil mineral that some plants incorporate into their tissues, but it's chemically the same as glass. So basically what this plant is doing is it's absorbing silicon dioxide from the soil, and then it's using that to reinforce these trichomes. So basically what we have are glass hypodermic needles that stab into your skin and then inject venom when they break off inside of your skin. This is why stinging nettle is so irritating. Not only is it, this hollow glass tube full of venom, it's breaking off the side of your skin. So that's why it's so irritating. We're going to switch now to looking at the internal anatomy of a stem. So internal stem anatomy is super important, I think, to understand like how the actual growth and development processes happen in plants, because we're really looking at, at the stem tips, we're looking at the formation of new organs like new stem, new leaves, new flowers, etc. Inside stems, we have a couple main structures. We have meristems. Meristems are areas of active cell division where growth occurs. And we've got a couple different types that we'll look at. We're not going to get that deep into secondary meristems in this video. There'll be another video later about woody stem anatomy, where we discuss things like vascular cambium and cork cambium. In this video, we'll be mostly talking about primary meristems, like apical meristems, axillary meristems, and intercalary meristems. Primary meristems, they make the stem grow longer. Secondary meristems make the stem grow thicker. We'll also be discussing transport tissues and vascular bundles. That's basically how does a plant transport resources such as water or sugars around their body. We won't get too into fiber cells. That'll probably also get saved for our my video on woody stem anatomy and also ground tissues. I'm not going to talk probably about things like pericycle and cortex in this video, just for brevity's sake. But ground tissues are essentially the stuffing of the stem, and we'll discuss that a little bit more in detail in a minute. Okay, the kind of first and most, arguably the most important internal part of a stem is the apical meristem. So the apical meristem is found at the apical bud, at the tip of the stem, and like I said a second ago, meristems are tissues which are undergoing active cell division and primary growth at the tip of a stem. So active cell division means that those cells are kind of rapidly dividing, and that requires the cell to duplicate all of its organelles, and it requires it to duplicate all of its DNA and then split in half and form two daughter cells. Primary growth means that the stem is getting longer. So the apical meristem is the only place where this primary growth occurs. And every new organ system of a plant is, I should say, like typically in most plants, every above ground organ system, such as a leaf or a flower or whatever, they're all being formed at this apical meristem where active cell division is occurring. That's where new skin of the plant or new epidermis is formed. That's where new leaves are formed, new buds, new vascular tissues, other internal stem tissues, etc. And because this apical meristem has to be able to develop into so many different types of tissues, I think it's valuable to think of this as like permanent embryonic tissue, or maybe you're familiar with the idea of stem cells. A stem cell is a cell that hasn't differentiated into a particular specialized type of cell yet. So when we think of an adult human body, we think of muscle cells and liver cells and nerve cells, neurons, and maybe a bunch of other types of cells, bone cells that make up specialized tissues. Um, an embryo of an animal is not composed of specialized cells, especially in the early stages of embryo development. They're just human cells. They're very generalized cells, and they have the ability to then divide and divide and divide and eventually differentiate or specialize into particular types of cells by either turning on or shutting off some of their genes. This is the same condition that we have in the apical meristem. This is essentially embryonic tissue. It is just plant tissue. It can develop into a leaf, it could develop into a flower, it could develop into more stem. And so I think it's helpful to think about it as basically embryonic tissue that's constantly dividing, and it's not highly specialized yet. And at the apical meristem, we're seeing primary growth. So primary growth is basically just growth that makes the stem longer. And one of the most frequent misconceptions that I'm really going to try to drive home through my discussion here is that a lot of people don't understand this, and then it causes problems for them. Longitudinal growth or lengthwise growth in stems only occurs at those soft apical meristem shoots. It does not occur in mature stems. So once a soft new shoot has gotten kind of hard and stopped elongating, that's it. It does not continue to get longer in that older mature section of the shoot. Only the tips grow. So when we look at like this red tip Photinia, Photinia x fraseri is the Latin name if you're curious on that. You can see that the older woody stems are kind of green, and then the new shoots are red, and that's where we have kind of active growth. Once those shoots harden up, they will not continue to elongate anymore. It'll require the production of a new burst of growth from the tip of the stem in order for that branch to continue growing. Or if we look at this huge old oak tree on the kind of bottom right corner, those large branches that are emerging from the trunk about 6 feet above the ground, they're that trunk is not going to get longer. That trunk is the length it will be. And if the tree wants to grow taller, it's only growing taller from the tips of the branches. This is, I think, one of the reasons people get confused about plant growth is because this is very unlike the way like animals grow. So, like bones, for example, grow at two growth plates at either end of the bone, but it's actually this plate that is maybe an inch or two from the end of the bone in large bones. And that's where cell division occurs that makes the bone longer. So the bone is actually getting longer from the kind of the inside out. Or if you look at nails and hair, the only place where they have new cells being produced is at the base of the nail or the hair follicle that's causing it to kind of grow and push out. And that's basically the reverse of what we see in plants. In plants, it's that new soft tissue at the tips of the stems that grows. Once the tissue has elongated, it no longer continues to get longer. At the very, 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 very tip of the apical meristem, we see the region of division. And in this microscope slide, you can see um, that these cells are really, 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 really tiny. They're packed together very, very densely. They're very small cells, and they're small because they're dividing so rapidly, they never really finish growing into a mature cell. That's part of what keeps it embryonic um, and able to develop into so many different things. So these are tissues of the apical meristem that are undergoing rapid cell division, and they are forming all of the different new types of tissue. So in this region of division, you can see there's kind of this dark point at the tip of the stem, at the shoot apical meristem, and then on either side we have two tiny little baby leaves that are growing. And basically, this process will just happen over and over again. The stem will grow a little bit, it'll produce a set of baby leaves, it'll grow a little bit more, it'll produce a set of baby leaves, and that basically establishes each node and intranode of the stem. Right after those cells divide, because they're continuously dividing in an actively growing stem, some of the cells kind of get pushed out of that active region of cell division, and they actually undergo growth. This is called the region of elongation. So this is a region in which those newly divided cells from that region of division at the very tip of the apical meristem, they inflate with water, and that is going to result in stem elongation and growth. And once an internode has elongated, it no longer elongates anymore. It's only capable of elongation during that early developmental stage, then it stops and it's done. Um, so you can see in this microscope slide that at the very tip of the stem, those cells are really, really small and densely packed. And then sequentially, as we move down the stem from that apical meristem, we can see that the cells progressively become larger and larger and larger. And that's because the cells farther down have filled with fluid, which has caused the stem to elongate. So the region of elongation is where that actual primary growth or lengthening of the stem occurs. Once the tissue has fully elongated, the cell walls start to form. And once the cell walls have formed around all those soft new cells, the tissue hardens and it becomes rigid and incapable of further elongation. Um, we've already kind of briefly introduced this idea, but at the apical meristem, we produce new organs. We produce new leaves, new buds, and new flowers, and these are referred to as primordia. A singular could be primordium, so we could talk about a leaf primordium or the leaf primordia. Um, these are basically kind of baby organs. So a leaf primordia is basically like a little baby developing leaf. So looking at this microscope slide of an apical meristem, we can see on either side of the apical meristem itself, we can see two little baby leaf primordia that are tiny itty bitty new leaves that the stem tip is producing. And if we look just outside of those, we can see these much larger, although still tiny, leaf primordia, which are kind of actively growing and expanding and eventually will be, um, large enough to be normal looking leaves that we could see with our naked eye. At this stage, when they're developing the apical meristem, they're typically not visible to the naked eye, and you often need a microscope in order to see them. We also see the development of bud primordia. So if we find our shoot apical meristem and then look down to either side, there's these other two really, really dark, dense spots in the axils of the older leaf primordia. Those are our bud primordia, and those will develop into our axillary buds. And the apical bud is also, or the apical meristem is also capable of producing floral primordia. So those are basically floral shoot buds. So remember that a flower itself is just a modified shoot, and all of the floral whorls or floral organs are just modified leaves. So the calyx is a set of modified leaves at a node. The petals are a set of modified leaves at a node. The stamens are a set of modified leaves at a node. And the carpels are a set of modified leaves at a node. So this is basically just a specialized bud primordia that, due to a combination of environmental and hormonal cues, develops into a specialized floral shoot rather than a vegetative shoot that would produce leaves. And you can see kind of in this diagram how that process occurs. Um, the developing bud primordia can either develop into a vegetative bud, an axillary bud, or they can develop into a floral bud. And kind of at the beginning, they're the same, but then, like I said, due to environmental and hormonal triggers, the bud kind of chooses one way or the other to go. And you can see, if you're curious, in this bottom image, kind of two different depictions of how this process occurs. One is showing the bud primordia from an electron scanning micrograph. So we're actually seeing the three-dimensional structure of that bud as the different floral whorls develop. And then in the image on the right, we can see cross-sectional images of that developing floral bud from its most basic stage, where it's just producing kind of the original base of the flower, till we see the production of calyx along the top row, and then we can see the production of petals in the second row and the beginnings of stamens. And then on the very bottom row, we start to see a pistil with an ovule and some stamens and petals and calyx all present in that very bottom right image of that right hand panel. The apical meristem is capable of a number of different things which are horticulturally significant, and I'm going to try to go through them as best I can. The first is fasciation. So normally our apical meristem forms a single point of growth, and that will result in a cylindrical shoot. But sometimes, rather than growing as a single point of growth, that apical meristem kind of flattens out and becomes linear. And then it produces this fan-shaped shoot instead. And this is a deformity called fasciation. If you have ever had plantar fasciitis, or if you have ever heard of the plantar fascia, that's a sheet of connective tissue that runs along the bottom of your foot. It kind of bridges your arch. And a fascia is really just a sheet. So fasciation is basically our apical meristem becoming a sheet-like stem rather than a normal cylindrical stem. Plants that exhibit fasciation are often referred to as cristate, which is basically a fun Latin word which means crested. And some of our ornamental plants are actually specifically bred for or propagated on the basis of having this deformity because it can look kind of cool. Celosia is one, or it's also called coxcomb because of the resemblance of the flower to the comb on top of a rooster's head, which has a fasciated inflorescence. And also there are some cristate or crested cacti, which have a linear apical meristem, which causes them to grow in this fan-like fashion. And the fun thing about this fan-like form is eventually this linear meristem kind of starts running out of space, and so it'll kind of continuously elongate and start folding on itself. And so sometimes these cristate cactus, when they're really mature and large, they kind of look like a giant brain because they're so convoluted and folded. When you see fasciation on landscape plants, it is kind of cool. You can see in this bottom right corner, there's a fasciated stem of some kind of landscape shrub. But it's usually best to remove these fasciated shoots. They don't tend to look very good, and they don't tend to produce normal growth after that point. So the best thing to do, in my experience, is just find the go back all the way into some definitely normal tissue, and just prune that branch off all the way back into some normal tissue, so that you get some normal stems growing back, that will tend to look better. Fasciation is something that is desirable in some plants, but in others, it just doesn't really look that good. It's more of just a curiosity. Another thing that's really horticulturally significant about the apical meristem is the propensity of rapidly dividing cells to sometimes make errors in DNA duplication. When you have hundreds or thousands of tiny little cells packed together tightly that are constantly dividing, they're having to duplicate their entire genome, all of their DNA, every gene in the genome, um, hundreds or thousands and thousands of times over the life of a plant, especially a large plant such as a large conifer, for example, in order to produce all those new tissues. So what can happen is inevitably in the thousands and thousands and millions of cell divisions that are required in order for a plant to grow from a seedling to a mature tree, sometimes those cells can make a mistake duplicating their DNA. And that results in a mutation, a change in the genetic code, which can sometimes go totally unnoticed. It might be a gene affecting, like, I don't know, drought tolerance or something. And if the plant's getting enough water, it doesn't really, you don't notice anything different. But sometimes we get kind of a drastic change in the phenotype or the gene expression, the appearance of the shoot from that point onward. And we call these sports. So a sport occurs when the daughter cells of a vegetative mutant cell, so a cell that was produced by faulty DNA duplication in those rapidly dividing apical meristem cells. When we get the daughter cells of that mutant cell becoming the dominant cell population in the apical meristem, from that point onward, the shoot will have an altered or a different phenotype. It'll appear different, obviously different, and you can see that in this upper image on this blue spruce. So we have this spruce which is surrounded by a bunch of normal blue spruces, and at its apical meristem, clearly there was an error in DNA duplication in a cell in that apical meristem, and then the daughter cells of that mutant cells, they became the most common cells in that apical meristem. They kind of took over, and then from that point on, we only had dwarf stem growth rather than normal stem growth. And this is where we get our dwarf blue spruce cultivars that are popular landscape plants. Um, if you're a skilled horticulturist, might notice that in a forest and say, "Oh, that's really cool," and then collect a shoot and either by layering or maybe a cutting or maybe by grafting, and they're going to propagate that in a nursery and maybe patent it and then sell it. And I really can't emphasize enough that a huge number of our new cultivars originate as sports, especially in our nursery crops. A lot of the floral crops, there's a lot of breeding that goes on, but because woody landscape plants have such low reproductive rates, because they take so many years to grow from a seedling to a mature plant, breeding programs tend to be very slow. And so often it's faster to use sports to discover kind of improved or more attractive or interesting new traits, to develop new landscape plants rather than through traditional plant breeding. Um, Sometimes we end up with a vegetative mutation, but the vegetative mutation decides to play nice with the existing normal tissue. And so we end up with both the mutant tissue and the normal tissue living alongside each other. And we call this a chimera. So a chimera is just an organism composed of cells of two or more genotypes, or basically two or more different, genetically different populations of cells. In plants, these are due to vegetative mutations. So we had all normal cells at one point, and then somewhere along the road, we ended up with a, a mutant cell due to an error in DNA duplication that the daughter cells of that mutant cell multiplied, but they never really took over. They just decided to kind of chill out and also continue undergoing cell division with our normal cells. Um, in animals, we can also have chimeras. They're not super common, but they're also not super rare. A chimera in animals is kind of like the opposite of a twin. So when we look at like what do we call monozygotic twins or identical twins, we have one egg that's fertilized by one sperm, and then rather than developing into an embryo in that first cell division, the embryo actually, or the fertilized egg actually splits into two separate um, fertilized eggs and then continues multiplying and forms two separate embryos that are genetically identical. Imagine instead that we have two different eggs which are fertilized by two different sperm. So we have two eggs that are released at the same time. So maybe we would have gotten what we call fraternal twins or dizygotic twins, but both of the eggs actually unite into one embryo. Um, so this does occur. There are human beings that have two different populations of cells that are genetically different. Um, in some cases, we can even have people who have um, maybe one set of XY or male cells that fused with some female cells that are XX, that were two separate fertilized eggs that combined, and that can result in a variety of different intersex conditions as well. So a chimera is basically just an organism composed of cells of two or more different genotypes. These occur in different ways in plants and animals. In plants, they're due to vegetative mutations. In animals, they occur due to the fusion of two different fertilized eggs during those early stages of embryo development. And in plants, a lot of our ornamental variegated plants are chimeral. So if we look at this philodendron pink princess, you can see that there are cells which are pink and there are cells which are green. And the cells which are pink are producing anthocyanins or red pigments, but they are not producing any chlorophylls. So they're basically chlorophyll albino cells. They have defective genes for the production of chlorophyll, but they're still capable of producing anthocyanins. So rather than getting this dark green leaf, we get kind of a mix of dark green and pink. And there's a couple different ways that these chimeras can occur that account for our different variegation patterns. In order to understand variegation and chimerism in the apical meristem, I think it's very important to talk about the tunica corpus theory. This is basically a theory of tissue development. It explains how tissue development occurs in the apical meristem, and understanding tunica corpus theory is really important for understanding how to maintain and influence um, ornamental variegations in horticultural plants. So the tunica, so this basically just means the same thing as tunic, right? A tunic is like a long shirt or a short dress that forms I should say formed the basis of clothing in a lot of different cultures throughout time. So tunica is just kind of a covering or the outer tunic or covering of the apical meristem. And this is composed of two different layers. There's layer 1 or L1, which develops into the epidermis, and then layer 2 or L2, which develops into our superficial internal tissues that are just inside the skin. And then under that, we have the corpus. So this is called L3 or layer 3, and it can be composed of one or more different layers of cells. And this develops into deep internal tissue, so like the very middle tissues of the leaf or the deep tissues of the stem. Um, Paraclinal chimeral variegations are a type of variegation where the daughter cells of an original mutant cell populate an entire tunic, typically L2, and they result in um, this entire layer of the apical meristem being composed of mutant cells. So the L1 is normal, L2 is mutant, and then L3, the corpus is normal. Um, so um, these were typically periclinal variegations result in a white or yellow leaf edge when the mutation is an albino mutation. Albino meaning no production of chlorophyll. We've got some kind of mutation in the genetic code of those mutant cells that means they don't produce chlorophyll properly anymore. So um, those mutations are very attractive. But because this is chimeral, because it's due to a vegetative mutation in the apical meristem, it's not typically heritable. So a variegated plant is not necessarily going to produce variegated offspring if you grow them from seed. So typically, variegated plants have to be propagated vegetatively through cuttings because they will not produce you have no guarantee that they'll produce variegated seedlings. A reverse periclinal variegation is when the L3 or the corpus is composed of mutant albino cells, but L1 and L2, the tunica, is composed of normal cells. And when we see a reverse periclinal chimeral variegation, we see the opposite pattern. We see a white or a yellow leaf center with a green edge. And these mutations are also relatively stable, like our periclinal variegation, so they're very common in horticultural plants. Um, and I'll talk about what stability means in just a minute. Basically, it means that they tend to stay that way for a long period of time without either over variegating or reverting back to green. Mericlinal variegations are super unstable and they're not really horticulturally common. Usually they're kind of more of a novelty or like a, ha, look at this surprise that happens every once in a while. Um, they result when an original mutant cell's daughters take over just one portion of one tunica layer. And they're super unstable because of that, because that tunic tends to kind of just continue to grow out and eventually repopulate with normal cells. And then we don't get to keep that interesting variegation. Sectorial variegation is when the daughter cells of a mutant cell populate all of they populate a portion of all of the layers of the tunica and the corpus. And because they take over just one portion, they aren't super stable variegations, but they are very attractive. They tend to be chunky, more speckled variegations rather than kind of a symmetrical. They tend to be asymmetrical patterns rather than being symmetrical like our periclinal and reverse periclinal variegations. You might be familiar with some of the variegated monsteras or the variegated philodendrons or even variegated Japanese maples that have these speckling patterns or patches of color, chunky patches of color over the leaf. Um, they are unstable, so plants that exhibit sectorial variegations are very likely to either over-variegate to kind of an entirely white or an entirely yellow or an entirely pink leaf, which is a problem because that means that all of the tissue from that point out is incapable of photosynthesis because it contains no chlorophyll and is therefore not capable of long-term growth. Or they can also revert to being completely green relatively easily, and that's obviously not super desirable either. So these plants do require a little bit more work to maintain the variegation because they're unstable, but they are attractive and they are present in commerce in the horticulture industry. Um, Variegation reversion occurs when all of the layers of the tunica and the corpus are repopulated with normal cells. So you might have a variegated plant in your yard, and then it throws a shoot that is entirely green and not variegated at all. That just means that the normal cells have retaken over all of the layers of the apical meristem with those normal green cells. And in order to maintain the variegation of a plant, you would want to maintain the variegation by pruning out any reverted stems all the way back into normal variegated tissue. The reverse can also happen where we over-variegate when all of the layers of the apical meristem become populated with the mutant albino cells, and then you end up with leaves that are entirely white or entirely yellow. And this is also undesirable because they're going to tend to not grow super well anyway, because they can't photosynthesize on their own. So I would say that reversion is more of a problem than over-variegation. The over-variegated shoots, because they can't photosynthesize on their own, they tend to not grow too much. But the green shoots are actually better at photosynthesis than the variegated shoots because they have more chlorophyll. So they actually tend to kind of outgrow your variegated material. And in some advanced cases, you can kind of swamp and shade out all of the variegated material of your plant until there's only green material left, because it's kind of outgrown and shaded out all your variegated shoots. So with any variegated plant, it is important to maintain that by periodically removing any reverted or variegated tissues all the way back into your desirable variegated tissue. Um, we do have some other types of meristems besides the apical meristem. We also have axillary meristems. These are the dormant primary meristems found in each leaf axil. Primary meristem, remember that that means that it's capable of elongating stem growth. So in this case, these axillary meristems can develop into axillary buds, which can then sprout and develop into lateral shoots or side branches or whatever you want to call them. Intercalary meristems are really unusual. You may remember that I just went on and on and on about how growth only occurs at the tips of stems. And that's true for almost all plants, but not for some of the grasses, bamboos, and monocots. In an intercalary meristem, above each node, we have this soft tissue where active cell division and elongation can occur, kind of at the bottom of each internode, just above each node. And that allows grasses and bamboos to do some really, really interesting things. So bamboos are really a family of giant woody grasses. So I guess saying grasses and bamboos is probably a little silly, but um, because of the intercalary meristems, grasses and bamboos are actually capable of elongating multiple internodes simultaneously. And this allows for extremely rapid growth. And in the case of grasses, which are living on grasslands, typically we see that intercalary meristems allow grasses to recover very rapidly after fire or grazing or other disturbances, which are very common on grasslands. Um, and in bamboo, it allows shoots to grow extraordinarily fast. So if you look at this bottom image, if you look at these new bamboo shoots which are emerging from the soil, you can see the scaly leaves which are covering the outside of that shoot. And we know that each leaf has to be attached to the node. So if we count the leaves, we can figure out how many nodes are underneath the leaves on this shoot. I count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 little leaves on this big kind of central shoot that's the most prominent in the image. When we look at this at a mature bamboo shoot, at the same height, we only see 1, 2, maybe 3 nodes, right? So what that tells us is that the nodes actually elongated after they developed. So um, the cell division and growth isn't just occurring at the apical meristem; it's also occurring in these intercalary meristems. And because the bamboo can basically preform all of its nodes, but then still do primary growth at the intercalary meristem, it allows the bamboo to basically do all this prep work, developing this young bamboo shoot that has a bunch of nodes. And then all of the nodes can simultaneously elongate. And that allows those bamboo um, shoots to just shoot up incredibly fast. Some species can grow over a foot a day because they're able to simultaneously grow at all of those different intercalary meristems, which is really impressive. Secondary meristems we'll be discussing more in depth in a coming video about woody plant anatomy, but these are meristems that allow for secondary growth or thickening of the stem, and they're often referred to as cambium. Um, we're not going to go into a lot of depth. Most herbaceous plants, meaning plants that do not have woody stems, undergo very limited to no secondary growth, whereas plants which are woody often undergo substantial secondary growth. So we'll be just diving into that more in a coming unit. Um, Transport tissues are extraordinarily important when we are looking at the physiology of stems. These are specialized tissues which have the ability to move water, mineral nutrients, food, and anything else the plant needs to move between the roots and the leaves. And there are two main types of transport tissues, xylem and phloem. And I'm going to do my best to make sure that we learn how to differentiate between xylem and phloem and remember which one does which, because the names often sound like nonsense. And a lot of people have, they might learn that one does one thing and one does the other, but they often mix up the names. So we're going to try to make sure it's really, really clear by the end of this video. The first transport tissue we're going to discuss is the xylem. So the xylem transports water and mineral nutrients from the soil up the stem from the roots to the leaves. So xylem is involved in moving soil resources up, and the soil resources that plants are collecting are water and mineral nutrients. And the cells that make up active xylem channels are dead. You can imagine that if you had living cells that were packed full of organelles and nuclei and endoplasmic reticulum and ribosomes and all kinds of other things, all those organelles would get in the way of the flow of water. So after these new xylem channels form, the plant actually kills off the cells and reabsorbs all the contents to basically create this empty pipe that allows for the water to flow up the stem with no obstructions. These xylem tubes are only 1 cell thick in diameter, and they basically, all they do is carry water and mineral nutrients up the stem. The mnemonic that I like to use to remember is that X is at the bottom of the alphabet, and xylem, which begins with an X, starts at the bottom of the plant, at the roots, and carries water and mineral nutrients up. Phloem, phloem, rather than transporting what I'm going to call soil resources, phloem transports photosynthesis resources. So we've talked a lot about photosynthesis in our video about leaves, and we know that the product of photosynthesis is sugar or glucose. But the fun bit is once plants have carbon available in their cells as a sugar, they can actually chemically react those sugars with other things, or but they can break them down partially, or they can add little bits of other chemicals, and they can make basically whatever they want. So those could be like insect repellent chemicals. Those could be, maybe starches, whatever the plant wants to make with those sugars. Phloem is used to transport sugar and the other things that the plant has made with those sugars up or down the stem to anywhere that sugar or photosynthetic product is needed. If we think about the roots are underground and they typically cannot do photosynthesis because they're not exposed to light. So the roots have to receive sugar or food from the leaves in order to continue to live and grow. So the phloem transports sugars made in the leaves down through the stem, through the roots to those growing root tips to feed them and continue root growth. But sometimes we might need to move sugars to the end of a branch. So for example, if the plant has flowered in the spring and now it's developing a large crop of fruit, it might need to move the sugars up the stem to a tip of a stem in order to fuel the growth of a fruit and fill it with sugar so that it becomes appealing to an animal, which will eat it and then help disperse the seeds of that plant. And that sugar can also be transported up to the tips of the shoots to fuel new growth, especially in the spring when a tree is maybe first leafing out for the year, or even an herbaceous plant that is kind of sending up its first shoots in the early springtime. The cells that make up phloem are actually alive. The flow of water in the xylem has to occur pretty rapidly because plants use a lot of water. The sugars the plant can afford to move a little bit more slowly. And so the cells which make up phloem are actually alive. They don't have to be these empty tubes that can transport large amounts of water really quickly. That's another difference between xylem and phloem. And the mnemonic I like to remember is that sugars phloem down from the leaves to the roots. Okay, which is, if you're not following along, it's a play on words with flow, to flow down. So the sugars phloem down from the leaves to the roots, and that'll help you remember that phloem transports sugars, not water and mineral nutrients like xylem. Um, most plants don't just have xylem and phloem randomly scattered throughout the entire stem. They have the xylem and phloem organized into what are called vascular bundles. And these are basically just clusters of these transport tissues. So the plant has, a little cluster of these xylem tubes, which are just one cell thick, and they're formed basically by cells stacked end to end. And the same thing with phloem; it's got them all kind of bundled together. In within a vascular bundle, we have kind of an interesting pattern. We almost always, at least in the case of our dicots, we have xylem always on the inside and phloem is always on the outside of a vascular bundle. In the monocots, it's a little bit um, different. We often have kind of xylem on the inside of the bundle and then phloem on the in the middle of the bundle. It's kind of weird. Monocots have weird stems. Um, One of the more useful differences, I would say, in identifying kind of differences between monocots and dicots is that dicots have vascular bundles which are always arranged into a ring, whereas monocots will have vascular bundles which are scattered throughout the entire inside of the stem. So when we look at a maple tree, for example, if we were to cut that trunk open, we would actually get growth rings because maples are a dicot, so they're going to have their vascular bundles arranged in rings. If you were to cut open a date palm or a coconut palm, we would actually not find any growth rings because palms are monocots with scattered vascular bundles. So the bundles are capable of thickening from the inside, but it doesn't produce any growth rings. You can't determine the age of a plant like a palm by looking at the inside of the trunk because there are no growth rings. Um, So we've talked about the epidermis of the stem. We've talked about our vascular bundles and xylem and phloem. Um, I could wax long in the tooth about ground tissues, but these are basically a variety of other stem tissues that are not vascular, meaning they don't transport material, and they're also not dermal, so they're not skin tissue. And this is often kind of thought of as the filling of the stem. There are several different kinds, and they do have specialized functions, but it's kind of beyond the scope and practicality of this course to get into that in great detail. One that's useful to know about is pith. This is a spongy ground tissue that's found in the center of the stems of a lot of plants, especially dicots. And you can see that this dicot stem on the left-hand side has the whole inner portion of the stem inside of those vascular bundles um, filled with pith, spongy tissue. We can also have stems can be modified in a huge different array of ways. We're going to try to get through these pretty quickly. We've already covered the major anatomy, so I would consider this second part to be mostly, um, if you're curious, understanding our different types of modified stems is very useful for propagating plants, which is why it's included here. Rhizomes are horizontal stems at or under the soil surface, and they help to spread the plant horizontally through the soil. So a lot of plants will kind of form a mat of these stems at soil level and kind of grow into this big clump. Um, some really good examples of rhizomes would include irises. In this bottom image, you can see irises, or something like ginger in this top image. So often people call it ginger root. We can see that there are little white roots emerging from these rhizomes or these stems, but we can tell that they are stems because they have nodes, they have lines at each node, and they have buds. So if they have nodes and buds and internodes, they must be stems and not roots. So the roots are the little white hair-like structures. The thick, white, fleshy organs at the base of this ginger plant in the top image, those are actually modified stems called rhizomes. Um, Bamboos and grasses often have rhizomes, which they use to kind of spread themselves underground, but they also have a second type of stem called a culm, and the culms are the above ground stems of grasses and bamboos. And culms are kind of specialized in a couple ways. They're hollow at each internode. They have a wall. Sorry, at each node they have a wall that kind of separates the chamber of each internode from the next one. Um, and it's useful to be aware that there are both types of stems. The rhizomes do periodically at their nodes produce lateral shoots, which become the culms. And one of the unique things about these bamboos is the rhizomes tend to be the permanent stems, and the culms tend to be somewhat temporary. The above ground stems of grasses and bamboos tend to have kind of a lifespan. They live for a while and then they start looking not so good, so you can cut them down, and then the rhizome under the soil will just send up more culms to replace any worn out culms. So yeah, the rhizomes are more of the permanent stems, and the culms are somewhat temporary in grasses and bamboos. Anyone who's cut open an onion will be familiar with a basal plate. So this is a compact stem found at the base of a bulb. And if you've ever planted tulips in your yard, you may notice that what was maybe one original bulb will eventually grow into kind of a clump of tulips rather than just one single tulip. And that's because bulbs are capable of propagating themselves by producing bulblets. And those bulblets are just small new bulbs that form from a lateral bud of the basal plate. So even though the stem is basically completely squashed, it does have leaves, which are the scales of the bulb. It does have nodes where each leaf is attached. It does have an internode between each leaf attachment, and it does have axillary buds that are capable of producing bulblets. And some examples that we're all familiar with: onions and garlic, tulips, daffodils, lilies, and amaryllis, which is a popular houseplant. In the case of garlic produces mostly lateral bulblets. It doesn't really have a primary bulb, and we call those cloves. The cloves of garlic are all little bulblets. Forms are often mistaken for bulbs, but these are actually the swollen base of a stem, which is used for food storage. And we can tell that corms are stems because they have nodes. They have lines on them where they can produce new lateral buds, and those lateral buds are called cormels or new corms. Um, so you can actually break off the cormels and plant them separately, and they will grow into a new corm, which is a really easy way to propagate cormous plants. Good examples would include crocus, which is on the far bottom left-hand picture, the purple flower, which are very popular early spring flowering corms. Calla lilies in the middle, Zontodacea, and then gladiolus, Watsonia, and crocosmia, which are all popular as garden and cut flowers. Tubers are very thickened underground stems, which are specialized for food storage. And they're a little bit different from corms because corms kind of stack on top of each other. A tuber tends to grow more or less continuously. It doesn't tend to produce like new tubers branching off the original tuber. And they are covered in nodes called eyes. So rather than having these kind of line-like nodes like a corm does, they have kind of separate nodes that we call eyes. And potatoes are probably the most familiar example. Anyone who's left potatoes in the cupboard for too long knows that they will start to produce lateral shoots from those nodes or eyes. And if you planted a potato in your yard, it would grow those shoots up to the soil surface. And then once that shoot broke through the soil surface, it would actually start to produce large expanded leaves, and it would grow a nice lush stem, and it would even flower for you. Um, but most of the time, people just break those shoots off and peel their potatoes and cook them up anyway. True yams are another good example. True yams are actually tropical vines that are native to rainforests, and true yams are not very common in American grocery stores. Most of the time, what you're buying in an American grocery store is actually a sweet potato, and sweet potatoes are actually in the genus um, Epomia, which are the morning glories, and they're not actually technically a true tuber. They're a tuberous root. So a sweet potato is actually a root. A potato is actually a stem. A really popular horticultural plant that also has a tuber are the cyclamens. This kind of large bottom image with the pink flowers. This is a cyclamen, and you can see that there's a large tuber at the base of the plant that has a bunch of roots growing out of it. Our tuberous begonias also have tubers, which form in the fall. And you can see I've included a little photo of the flower of a tuberous begonia as well. Um, Stolons are above ground propagative stems that produce new little plantlets, and they're also called runners sometimes. Stolons are a common feature of many horticulturally popular plants and a really easy way to propagate plants. So this upper image is, you might recognize this as a strawberry. Your mother plant will produce these long stolons, these red stems, which at each node, technically at every other node, they'll produce a little plantlet. And then once those plantlets root in, they actually will produce a stolon of their own that continues growing out and then produces another plantlet every other node and so on. And that allows, one original strawberry plant to grow and fill in maybe your whole flower bed. Some other good examples, a very familiar one might be spider plants. In this bottom right-hand corner, spider plants in the wild would produce stolons that ran along the soil surface, just like a strawberry. But in the case of our house plants, the stolons just kind of hang down and then their plantlets kind of accumulate. And that's where they get the name spider plant, because they kind of resemble a spider hanging on a line of silk. There are some other maybe less well-known houseplants like Alsobia or Apicia that also have stolons. And maybe a more familiar example would be mint as well, has these above ground stems which creep along the soil surface to spread that plant. Ortets, ramets, and gannets, I think, are a kind of a useful thing to know about. This is really a slide about suckers, but I want to explain the idea of an ortet, a ramet, and a gannet because I think it's useful in other horticultural contexts. So an ortet is an original mother plant which germinated from seed. So anytime you sow a seed of a plant, that is an ortet. If the plant is capable of propagating itself in any way, any little baby plant the plant produces is called a ramet. So this is a clonal offspring of the ortet or from another ramet. So for example, when we looked at our strawberries, the original plant, which maybe grew from seed, right? That's our ortet. And then its baby that grows at the end of a stolon is a ramet. And then the ramet itself can produce its own baby ramet and so on and so forth. On many species of trees, the root system can actually produce adventitious buds. So anytime we see the word adventitious in plant science, that basically means that we have a tissue growing from another tissue that is dissimilar to the tissue it's growing from. So, for example, if you have a root that grows out of a leaf, or if you have a root that grows out of a stem, or if you have a stem that grows out of a root, those would be adventitious growths. So if we have, for example, a quaking aspen, which we see in this top image here, the root system of a quaking aspen, like we see in this bottom right-hand corner, is actually capable of sending up adventitious shoots from the root system itself. And what that allows the plant to do, the original ortet, or the original mother plant which germinated from seed, is as she grows her root system out, she can send up new trunks from her root system. And that will allow one original plant to become an entire grove or an entire colony of plants. And that whole colony is called a gennet. Another term for a gennet is a clonal colony or a clonal organism. And that's basically a single vegetative superorganism composed of an ortet and its ramets. And the reason I say ortet with an asterisk is because sometimes the ramets continue to grow and propagate and continue existing long after the original ortet has died. One of the best examples of a gennet or clonal colony is Pando. Pando is a grove of quaking aspen trees in Utah that originated from one original seedling. Pando is this grove of aspen trees that I think the estimated age on that is up to about 20,000 years old now. About the time of the last ice age, I could be 14,000 years old. So there's a range of different age estimates. It depends on what techniques you use, but really extremely ancient organism And we know we can estimate the age of the tree based on the fact that all the genetics of every trunk are functionally the same. We can have some minor vegetative mutations occur over time, but um, the we know the rate at which quaking aspens are capable of spreading their colonies, and we know the rate at which pando spreads new ramets or new babies. And we can estimate that this plant's been growing for about 14 between 20 and 14,000 years old, 14,000 years. This single plant covers 108 acres, and the weight is estimated at 6,000 tons. So remember that a ton is about 2,000 pounds. So that's just an extreme amount of weight for that one organism. Offsets are also called pups. These are small little plantlets that grow from the base of the stem of a plant. So if you've ever had an aloe vera or maybe an echeveria, you'll notice that over time they'll produce little babies around the base. These are pups, and you can really easily propagate these plants just by pulling off the pups and potting them separately. Some other plants, such as like date palms or bananas, also produce offsets or pups, and they can be propagated in the same way. Just pull off the pup, pot it by itself, voila, got a new plant. A basal rosette is a compact modified stem in which the internodes do not elongate. So when you look at a dandelion, we can see that there's this whirl of leaves that appear to all come out of the ground. And we know that leaves attach to a stem at a node. So what that means about a dandelion is that the stem is actually super, super compact and the internodes are extremely short. Same thing in an aloe. Each leaf of this aloe is attached at a node, but the internodes just don't elongate. So you just end up with this tightly packed arrangement of leaves in a little circle. It's called a rosette. Um, Other good examples might be yucca or echeverias. So a basal rosette. A lot of weeds have basal rosettes as well. Oh, thorns. So we've talked about prickles earlier. If you remember that, it's been a minute. So thorns are different than prickles. If you remember, the prickle is an extension of the epidermis or the skin of the plant. Thorns are sharp, modified lateral shoots, and they end in sharp points. We can tell in the upper example, this is the stem of Gleditsia triacanthos, which is the honey locust. Wild honey locust trees are extremely thorny, whereas the ones we commonly grow in landscapes are actually selected for thornlessness. We can tell that this is a lateral shoot because it has nodes. It has itself produced little lateral shoots, which are themselves thorns. And you can see that all of the thorns in this upper image have grown from a node where there was a bud. So it's basically this cluster of little lateral shoots that themselves branch out and they end up ending in a sharp point. The whole thing dries out and dies, and that helps defend the plant from herbivores. Plants are very good at making a weapon. We saw in our unit about leaves that cacti can modify their leaves into a spine. We've seen that plants can modify their epidermis or their skin into a prickle, and they can also modify a lateral shoot to develop into a thorn. Some other examples besides honey locust, which is what we see in this top image, are citrus. In this bottom left-hand corner, you can see that where we should have an axillary bud at some of these nodes, we instead have a thorn, and that's because the axillary bud has grown into a lateral shoot that decided to turn into a thorn. And another example would be Kynomeles, the flowering quince, which is this beautiful red flowered plant. And you can see once again that there is a thorn emerging from that node just above the flower. So don't confuse thorns with prickles or with spines, which are modified leaves. Ooh, stem tendrils. So sometimes these lateral shoots actually develop into a little tendril. So that's a modified lateral shoot, which the plant uses to climb. So grapes, for example, they produce stem tendrils, not leaf tendrils. Plants can modify either their leaves or their lateral shoots to produce tendrils. It depends on the genus or the species. Watermelons also produce stem tendrils, so do squashes and passion flower. And you can see in this top image, passion flower, you can see the tendrils that it's using to attach to this wire screen on the front of a house. Twining vines are an example of a really, really interesting stem type, which is just does some really, really fascinating thing. Charles Darwin was one of the first scientists to kind of formally investigate this. And because time-lapse photography did not exist during his time, what he did is he actually set up a series of strings around the plant. And then he would, I think he had his son periodically check the change of the angle of the stem tip over time and see, which strings it was hitting, and then kind of monitor the growth of that stem around its support. This twisting behavior that you see in this GIF of this bean in the top image is called circumnutation. So that comes from Latin. Circum means around, and then nodum means knot. That's where we get the word node as well. It's a knot in the stem. This is a circular pattern made by a stem tip as growth occurs unevenly in that region of elongation. And most plants do some level of circumnutation. Circumnutation basically, it has a couple different functions. If the tip of the plant just grows straight up, if it hits something, it doesn't necessarily know the best way to get around the thing that it's run into. So maybe it's a plant sprouting underneath, oh, I don't know, like a rotting log or something, but maybe the log is itself laying on another log. So this stem tip just kind of grows up through the air and then hits this log and then doesn't know where to go. If the plant has been circumnutating or kind of spinning in a tiny little circle, it can kind of, the plant can figure out which way is the best path upward because it will have sampled an area where it's maybe more clear or less clear, and then it can grow towards the area that's more clear. In these climbing vines, they actually don't want to grow towards clear space. They want to find something to wrap around. So they've actually got extremely exaggerated circumnutation as they search for something to climb it. And then once they find that thing, once they make contact, then they continue to circumnutate, but in kind of a more subtle way to continue to feel that support and make sure that they're still climbing up it in the right way. Some good examples would include beans, convolvulus, which are bindweeds. These are the morning glory, the invasive morning glory weeds that you get in your lawn. True morning glories in the genus Epomia, which are often grown as ornamental plants, and wisteria, which is a woody vine, which are usually called lianas. Ooh, cladodes are one of the most kind of unusual types of modified shoots. So I want you guys to look to begin with at this shoot of Arabidopsis compared to a shoot of an asparagus. When you look at the Arabidopsis, we see our typical normal stem anatomy. So we see that we've got our primary shoot, we have a node, the node has a leaf, and then just above the leaf in the axil, we had an axillary bud, and that axillary bud has developed into a branch or a lateral shoot. When we look at the asparagus, we see a slightly different pattern. We still have the primary stem, we have a node, and then the node does have a leaf, but the leaf is tiny. It's reduced to just a tiny little scale. And then above the leaf in the axil, we had an axillary bud that developed into a lateral shoot, but it doesn't look like a normal lateral shoot. It's flattened and it resembles a leaf. In this case, these flattened photosynthetic lateral shoots are called cladodes. And cladodes have a limited growth destiny. So unlike most lateral shoots or most shoots in general, they do not have the ability to continue to produce new branches. So that lateral shoot doesn't have the ability to produce tertiary and quaternary branches. It just is a cladode, and now it has a limited growth destiny. So good examples would include asparagus, which we can see on the far left-hand side. Butcher's bloom or ruscus, which is in the middle, you can see that each ruscus cladode does have one node on it from which it produces a flower. And ruscus is really popular in the floral industry as a floral foliage. And then my favorite example of a cladode in this far right-hand photo is semele. Semele is a plant from the Canary Islands off the coast of Africa. It does not have a common name so far as I'm aware. And you can really tell that these are stems because along the edge of the cladode, we have a series of nodes, and from each node, the cladode is capable of producing a number of flower buds. And I think that's super neat. We see that the primary shoot is just this normal cylindrical shoot, and then the lateral branches are these cladodes, and then each cladode is only capable of producing flowers. Um, it's not capable of producing new lateral shoots from the nodes on each cladode. But semele is a really gorgeous plant that I really wish I could grow at some point. Phylloclades are also leaf-like stems, but unlike a cladode, they have an unlimited growth destiny. So a phylloclade is actually capable of producing new stem growth. It can produce a new phylloclade or it can extend its own growth. Some really good examples of this would be prickly pear or the pad cacti. In this top right-hand corner, you can see a pad cactus. Each pad is a phylloclade. It is a stem. We can tell it's a stem because it's covered in nodes. And from any of the nodes on each phylloclade, the phylloclade is capable of producing a new phylloclade. So unlike a cladode, which can sometimes only maybe flower and that's it, the phylloclade can continue to produce new phylloclades and it can continue to grow and branch out. Another good example that you might be familiar with are the Christmas cacti in this middle right-hand picture. We can see the sequential phylloclades of a Christmas cactus. Each phylloclade can produce numerous new phylloclades and continue to grow and branch. Um, a lot of the euphorbias, such as this pencil tree on the left-hand side, they also have phylloclades. So what look like leaves at the end of these branches are actually just leaf-like stems. Or the ribbon plant, Homolocladium, which is not a super common plant in horticulture industry, but you can find it occasionally for sale as a house plant. And you can see the nodes on the stems, the phylloclades of the ribbon plant very clearly because each one is marked by a line. And we can see that there are lateral shoots of more phylloclades emerging from that central phylloclade in that bottom right-hand image. Bulbils are really interesting. So we've talked about bulbs and their basal plates, and we've talked about bulblets. Bulbils are not the same as bulblets. For starters, bulbils form above ground, and a bulbil is basically a small, immature plant that develops from an aerial bud. So if we look at this left-hand image, this is a lily, a true lily in the genus Lilium. And where we should have an axillary bud, and I should say we do have an axillary bud, but the axillary bud has actually developed into kind of a baby plant of its own. And you can even see little aerial roots forming. So what's going to happen in the winter as the top of the plant freezes down is those bulbils are going to be shed onto the soil surface. They are going to root in and they are going to become their own independent plant. So bulbils, an important thing is that they form above ground from a bud, and they are shed from the mother plant in order to propagate the plant. Other good examples would include garlic. We can see that the inflorescence of garlic in this middle right-hand picture produces a large number of bulbils, which can fall to the ground and propagate the garlic. Other good examples would be bulbous bluegrass, which is a common weed in the top right corner, which instead of producing an inflorescence also produces just a bunch of bulbils. And also some agaves, after they're done flowering, will produce a lot of bulbils which can fall to the ground to propagate themselves. And then our last type of modified stem that we'll discuss today are pachycols. So pachycolous plants have thickened stems which are used to store water to survive periods of drought. So a really common, a really familiar example should be the columnar cacti, which are in the family Cactaceae. Um, there are some euphorbia species which also have this type of pleated pachycolous stem. But the euphorbias, which are often sometimes mistaken for cacti, are not really cacti. Euphorbias are the cactus-like euphorbias are mostly native to Africa, and they've kind of evolved they've convergently evolved the same appearance as cacti, but they're not truly cacti themselves. All of the true cacti are restricted to North and South America in the wild. Another good example, and a plant that I'd love to grow at some point, is the desert rose, Adenium obesum, and that's this pink flowered plant in the top right-hand corner. They bloom right as they're coming out of dormancy, then they grow some leaves, they photosynthesize for a while during the rainy season, and then once it dries out, they lose all their leaves and they live the rest of the year just off of their water reserves, and they don't really grow very much. So they're very slow growing. Adenium obesum, which is named, obesum meaning obese because of that very thickened stem. Another really popular plant in horticulture is the ponytail palm, which is in the bottom right-hand corner. This is a plant growing in some kind of tropical garden, somewhere where it never freezes. The ponytail palm is native to Mexico, and it has this very, very fattened bulbous stem base in which to store water. But more often in the United States, you see Bocarnia or ponytail palm grown as a houseplant. And then a really familiar example as well for anyone who's watched like The Lion King or the movie Madagascar will be the baobab trees. These are trees in the genus Adansonia. In this bottom right-hand corner, we can see a baobab tree, and you can compare the trunk to the people standing next to it. Baobabs have extremely thickened trunks that are full of water. And this adaptation is mostly found in woody plants that live in frost-free regions in the tropics or areas where it doesn't maybe get that cold for very long. You can imagine that if these plants tried to grow in a temperate zone where it gets very, very cold or in the Arctic, water expands when it freezes. And so if you store a lot of water in your stem, it's going to be pretty hard to keep it from freezing and expanding and basically splitting your stem apart. So this is a trait that's mostly restricted to tropical and subtropical plants. You don't see this often in temperate or Arctic plants, but it's certainly an interesting feature horticulturally that a lot of people find ornamental and attractive. Um, and then it can also be ecologically significant. So baobab trees, for example, they store a lot of water in their trunks, and because of that, their wood is actually unusually soft. Um, and so during periods of prolonged drought, sometimes African elephants will actually use their tusks to basically tear open the wood because it's pretty soft wood on those baobabs. It's super watery. They'll basically tear apart the trunk of the baobab tree and they'll actually eat the wood for the water it contains. And so that's one way that elephants will actually hydrate themselves during periods of extreme drought if there's no surface water available or no safe surface water available as well. So yeah, that's kind of interesting. Another feature of these pachycols is that plants that have them often have really thick protective bark or they will have spines. I don't have an image of these on the slide, but the Pachypodium species from Madagascar as well as the cacti have extremely spiny stems, and that basically helps protect them from desert animals, which might want to get at some of that water. You can imagine that storing a bunch of water in a desert, you're stockpiling a resource that everyone wants. You have to be able to defend it, or you're not going to be able to hold on to it. So, and that's the case with the baobab. Elephants just tear their trunks up like no business, no other, because they don't have any defense mechanism. So, something to consider. But certainly pachycols are really, really interesting, and a lot of people find them fascinating and collect plants to have them. All right, so that is all for today. I hope that you've learned something about stems and enjoyed this explanation of stem anatomy and physiology and modified stem types. Have an excellent day.